In this short video, we'll be looking at another use case for the GNPS dashboard, and that is looking at MS1 data. So while many labs now have access to tandem mass spectrometers and can therefore acquire data for MS2-based molecular networking, routine analysis in many labs, such as natural products labs, is still often performed on unit resolution MS1 mass analyzers, like single quadrupole instruments. However, proprietary software often makes it difficult to rapidly analyze and share these types of data between laboratories. So to address this, there is functionality in the GNPS dashboard to allow you to visualize MS1 spectra. As an example use case, we are going to look at the metabolome of the methylotrophic bacterium Methylorubrum extorquens, which used to be called Methylobacterium extorquens. So this organism produces chemical signals, quorum sensing signals called acylhomoserine lactones to coordinate group behavior. And Vorhold and colleagues previously showed that two of the signals that are produced by Methylorubrum extorquens are 2E7Z C14 homoserine lactone and 7Z homoserine lactone. We're seeing those structures right here, and they differ only in one double bond. These two chemical signals are actually only produced when this organism is grown on the single carbon substrate methanol, but not on the multi-carbon substrate succinate. So to demonstrate the GNPS dashboard, what we did was we grew up Methylorubrum extorquens on both methanol and succinate. We analyzed those data for the extracellular metabolome using a single quadrupole mass spectrometer in Agilent 6120 LCMS, exported the data in a common NetCDF format, and then uploaded it to Massive so that it could be visualized rapidly using the GNPS dashboard. So here you can see in the first data group, we have our triplicate methanol grown cultures, whereas in our second data group, the bottom data group here, we have our triplicate succinate grown cultures. So you can see these two quorum sensing signals have protonated masses of 308 and 310. And so what the GNPS dashboard allows you to do is actually generate multiple extracted ion chromatograms at one time. So we're going to generate extracted ion chromatograms for both of these protonated masses. And then for our instrument, we're going to set a tolerance of 0.3 Daltons. So if you scroll down here, you can see there's a lot of information available. But one thing that you can look at is these two extracted ion chromatograms. So we have our one for 308. So this corresponds to the quorum sensing signal 2E7Z C14 homoserine lactone, as well as the one for 310. So this would be for 7Z homoserine lactone. And in both cases, at very similar retention times, since the compounds are quite similar, you can see something that is only present in the top or methanol grown data group, but not in the bottom or succinate grown data group. So this one is coming off at about 25.9 minutes, and this one is coming off at about 26.3 minutes. So what we can do is actually change the retention time window up here to zoom in on this area. And if we come back down here now, you can see a zoom in on this particular retention time window. And although this shows that these samples were probably overloaded, what this can be useful for is if you go down here, you can see that peak areas are automatically integrated for all these extracted ion chromatograms over the retention time window specified. So in this case now, you can see very clearly for these triplicate cultures that this feature with a mass of 310 in this retention time window is only found in the top or methanol grown culture, but not in the bottom or succinate grown culture. And same for this one with a mass of 308. So these likely correspond to the two quorum sensing signals shown here, and verify that these are only produced in the methanol, but not the succinate grown condition. So I hope this was helpful in terms of showing you how you can rapidly analyze and also share data using the GNPS dashboard, even for MS1 data.